technologies, and variety of plants and animals. 9 national scientists, over 30 outstanding young scientists, and more than 18,000 graduates contributing to agricultural development. UPCA, now College of Agriculture and Food Science, or CAFS, has been continuously contributing to nation building through its instruction, research, and extension function. A mere 10 years after establishment, the college was able to start breeding works with the production of sugarcane varieties with high productivity to Fiji and mosaic disease. In the 1920s, the importance of agriculture was intensified and UPCA was recognized as neighboring countries began sending their students to Los Baños because of the high quality of education. In the 1930s, breeds with superior characteristics such as the Burkhala pig, Los Baños Cantonese chicken, and filament cattle were developed. The 1960s was a fruitful decade for research. Makapuno, a coconut cultivar often made into a dessert, was pioneered and perfected. Meanwhile, the Dairy Training and Research Institute was the first in the country to freeze buffalo semen, paving the way for artificial insemination of carabaos. Along with the establishment of UPLB in the 70s, the college received the prestigious Ramon Magsaysay Award for International Understanding, Asia's counterpart for the Nobel Peace Prize. In the same decade, the C4 plants were developed. The soil test kits were produced in support of the Masagana 99 program, and the mango induction technique was introduced. Continuing the production of quality research outputs in the 1980s, techniques to minimize post-harvest losses, the accelerated fish sauce manufacturing process, and the rapid test kit for the detection of pesticide residue on vegetables were developed. The 1990s saw many new varieties across species or crop types, including Sinta papaya, the first Philippine-bred hybrid, UPLB Gold, a durian variety, and Doña Amelita, a musaenda named after the wife of former President Fidel Ramos. The award-winning National Corn-Based Farmer Scientist RDE Training Program, or FSTP, was also established around this time and still remains as one of the university's primary extension programs. In the field of instruction, the book titled Integrating Sustainable Agriculture in the BSA Curriculum, the UPLB College of Agriculture Experience, in 1999. At present, the College of Agriculture and Food Science, or CAFS, continues to be the premier institution of higher learning in agriculture and food science, and remains committed to nation building through its instruction, research, and extension programs. In order to produce the best graduates, all of our courses are outcomes-based education, and continuous improvement of the curriculum is being practiced to meet the challenges of the 21st century. As testament of excellence, CAFS is constantly recognized by different organizations. In research, we continue to implement relevant studies with focus on the 12 priority commodities to address food security. Moreover, technologies are constantly developed to aid in responsible food production. Supplementing these are extension activities to disseminate the knowledge generated to improve and livelihood of the people. We believe that present technologies will future-proof our food systems. Some of our notable developments are IDTech, Coraginam, Plant Health and Pest Clinic, pest management tools and techniques, smarter pest identification technology or speed tech, snap hydroponics, drone technology, rapid immunofilter assay for virus indexing, nanofertilizers Fertigrow, Hormogrow, nanobiosensors, biochar. We at the College of Agriculture and Food Science will continue to promote and nurture a culture with innovative yet conscientious minds and cutting-edge skills to lead and serve the country and the region. We are one college with one goal towards sustainable food systems.
Good morning, everyone, and welcome to the Coffee Webinar Series brought to you by the College of Agriculture and Food Science, University of the Philippines, Los Baños, Laguna. This event is made possible through the coordination and planning of the Ad Hoc Committee with Dr. Leah Fernando Corpus as chair, headed by our Dean of the College of Agriculture and Food Science, Dr. Elpidio M. Agbisit Jr. My name is Jessa Magtoltol from the Agricultural Systems Institute, and I am your host for this morning. Together with me is Ms. Sheni Hilado from the La Granja Research and Training Station, who will be our moderator during the open forum. At this point, let me acknowledge our participant for this morning through the different social media platforms. We are live via Zoom, Facebook, and YouTube. Let me encourage everyone to please share our episode today so that it could reach a wider audience. Also, let me acknowledge the presence of our Associate Dean of the College of Agriculture and Food Science, Dr. Leah Fernando Corpus. Ma'am, good morning po. And muli, magandang umaga po sa ating lahat.
Good morning.
To ensure a smooth program flow, here are our virtual house rules. For our panelists, keep your microphones muted at all times if called or requested. For Zoom participants, during our Q&A, use the chat box or raise hand function if you wish to directly ask the resource person. The moderator shall acknowledge the participants to ask their questions. Once called, please state your name and your organization. For our Facebook and YouTube viewers, use the comment section during the Q&A. Also, only those participants who completed and submitted their evaluation form, which will be posted in the comment section, will be given a certificate. As a recap for the last episode of our coffee webinar series last Wednesday, November 15, 2023, Dr. Ernelia P. Cao presented the development of genetic markers for coffee varieties grown in the Philippines. To those who want to access the previous episodes through our Facebook page, hanapin nyo lang po ang UPLB College of Agriculture and Food Science. So, sabay-sabay po tayong makinig sa ating episode 3 for this webinar session with the topic titled Identification and Genetic Characterization of the Philippine Coffee. To formally open our program for this morning, without further ado, ladies and gentlemen, let's welcome Dr. Sherry B. Marasigan, the Deputy of the Agricultural Systems Institute. Ma'am Sherry, good morning po.
Good morning, Jessa. Good morning, everyone. To Associate Dean, Dr. Lilia Fernando Corpus, to our guest speaker, Dr. Miriam Baltasar, the Director for Research of the Cavite State University, Ma'am Eva Ginabo of LGRTS, to our colleagues from the university, and to our guests who are with us in Zoom from all over the country. So we are very happy to have you in today's episode 3 of the Coffee Webinar series of the College of Agriculture and Food Science. So, nagkape na ba kayong lahat? No? Alam ko lahat po ay hindi nabubuhay ng walang kape. Manghihina daw ang mga tao pag walang kape sa katawan in a day. So, coffee, we all know, is part of the Filipinos' culture of our everyday living. So, locally produced commodities in recent years are usually equated with sustainable practices, rural development, and improved sense of community. So, this is especially true to commodities grown and marketed by local communities such as coffee. So the coffee industry benefits the country with employment for coffee value chain players, and thus generating income and opportunities to maximize the increasing local coffee consumption, and syempre generating opportunities to elevate the competitiveness of Philippine coffee in local and global markets. So coffee, as we all know, is one of the major products of Cavite and one of the major projects of the research activities of Cavite State University. Pero medyo complex yung topic today, no? Because our speaker will be talking about identification and genetic characterization of Philippine coffee. So ako very limited yung alam ko about this. Hindi ko nga alam kung ito nga ba yung tamang mga variety, no? Like yung mga Robusta, Arabica, no? So I know we will be able to further understand about the characterization of coffee in the Philippines through Dr. Baltasar. So let us take this opportunity to learn from what CVSU has been working on in terms of coffee in the country. So muli, maraming salamat po at magandang umaga po sa ating lahat. Jessa.
Thank you, Ma'am Sherry, for that warm greetings and welcome to start our webinar today. And indeed, we are excited to listen and learn from our speaker. But before we proceed to our program, may I request our panelists to please open your camera for our quick group photo to be assisted by our tech team po.
Okay po. 1, 2, 3. Okay na po. Thank you.
Okay, thank you, Sir Arthur. Ayan. So moving forward, let me take this opportunity to introduce our speaker this morning. Our speaker is a professor from Cavite State University. She is also an alumna of the University of the Philippines, Los Baños, and finished her Master of Science and PhD program in genetics. She also headed the projects on the morphological, characterization, identification, and conservation of Philippine coffee genetic resources. There is also a lot of ongoing R&D projects in her pipeline, and some of them are the genetic differentiation of the Coffea liberica using SSR marker analysis and DNA-based authentication of coffee sold in the Philippine markets, and many others. She is also the lead author of the assessment of the genetic diversity of Coffea arabica coffee in the Philippines using SSR markers. She presented her various research at the national and international conferences. Because of her expertise, she is staffed by the different agencies as a resource person and provides technical services. She is the editor-in-chief of the Philippine Coffee Journal and serves as a reviewer on various proposals and scientific journal articles. At present, she is the research director of the Cavite State University. Let's give a virtual applause to Dr. Miriam D. Baltazar. Ma'am, good morning po.
Good morning po. Naririnig po ba ako?
Yes po ma'am, loud and clear po.
Okay, so thank you so much. Pwede po ba ako mag-share ng screen?
Yes po ma'am.
Saglit po. Oops. Ayun, nakikita na po ba? Or medyo...
Yes po ma'am, kita na po.
Okay. Ayan, so good morning po sa inyong lahat. Ano po, yung tanong kanina ni Dr. Marasigan, nagkape na po ba kayo? Ako po actually hindi pa nagkakape 'no, kasi ay baka ako'y kabahan sa talk na 'to, dahil ina-anticipate ko parang napakarami po atang katanungan. Last week I joined the Zoom, meeting 'no, medyo late po ako dumating dun sa talk ni Dr. Cao. But, natutuwa ako na meron pong post dun si Ma'am Lilia. Sabi niya ay tatanong natin ang mga questions na 'to sa, Tuesday. So, naisip ko po kasi noong, nakikinig ako sa talk ni Dr. Cao, sabi ko, "Ano pa kaya ang sasabihin ko?" Parang nasabi na lahat ni Dr. Cao. Kasi I'm also a geneticist, 'no po. However, yun nga nakita ko na, may mga katanungan pa din ang ating mga coffee stakeholders, 'no po. So hopefully ay masasagot po natin this morning, and sabi ko dun sa isang group of farmers ano po who I invited to join this topic kasi alam ko this would be very important to them. I promised them not to talk very technical. So I'll try to make this talk na kaya pong intindihin ng karamihan. Okay, so once again, thank you, Doc Lilia, for inviting me to share this topic. Actually, this is very close to my heart. Ano po? Identification and genetic characterization of coffee or Coffea spp. So, marami na pong na-discuss si Dr. Cao about the genetics of coffee. But this morning, there will be terms na uulitin ko, but in the interest of time, I'll try not to discuss what has already been mentioned last week. Okay? Oops, di ata gumagalaw. Aha, ah, ayan, okay. So this is the outline of my presentation this morning, introduction. And then of course, I'll just discuss other coffee species with economic importance and potential. And then conservation of Philippine coffee. And of course, the core of this talk is on the identification and characterization of coffee. Okay. So, this slide shows of the process of producing a coffee beverage. Ano po? Well, it's a very complex process, but it only tells us that the coffee we drink and enjoy every day comes from the coffee plant whose fruits or cherries are harvested and processed. It has a stimulating effects ano po, and because of the caffeine and other metabolites. And coffee is our way of commune. It's our medium of socialization and relaxation. Kaya minsan ano po, sabi natin, tara magkape tayo. Okay? Pag tayo ay nai-stress na. Okay. Ano nga ba yung taxonomy ng coffee? So, Coffea or the genus Coffea had two subgenus, Baracoffea and Coffea, where the species used for beverage belong. Di po? Baracoffea and Coffea eventually merged into one genus, that's Coffea, making 104 species. So, there is this species known as Silanthus. Kaya yung descriptors for coffee, it's descriptors for Coffea and Silanthus. So, Silanthus is also under the same family, Rubiaceae, which is closely related or resembles coffee. So, in 2011, 20 species of Silanthus were transferred to the genus Coffea. But okay. In August 2021, 6 new species of coffee were discovered from northern Madagascar. So currently, there are 130 species under the genus Coffea. Okay. What are the economically important species of coffea used as beverage in the Philippines? So, we have the Coffea canephora, or ito yung robusta. Hindi po siya Coffea robusta. Meron ding Coffea arabica or the arabica, and Coffea liberica variety de Weuvray or Coffea de Weuvray, ito po yung excelsa. At saka yung Coffea liberica, variety liberica, or ito yung liberica na tinatawag natin. So there are 26 NSIC registered varieties of coffee in the Philippines. Therefore, hindi po I find it odd ano po when we call robusta variety or arabica variety. So Liberica variety or Excelsa variety. Kasi po, that would give confusion. So there are many varieties of Robusta. For example, FRT1, FRT7, 11, 65, 101, and so on. And you are familiar with the varieties of Arabica. For example, Granica, Red Bourbon, Geisha, Mundo Novo, and many others. So, that's why to my students, I often tell them, mas maganda siguro sabihin natin na types of coffee, yung Robusta, yung Arabica, yung Excelsa, at saka yung Liberica. Okay? So, this figure emphasizes that coffee is produced predominantly by countries along the coffee belt. So when we say coffee belt, this is found 25 degrees north and 30 degrees south of the equator with tropical and semi-temperate weather. Okay? So the figure below shows us the consumption, the imports and production of coffee in the Philippines. So as you can see, no, our consumption is far above our production. And to meet this demand of consumption in the Philippines, we needed to import. So in fact, more than 90% of our coffee today are being imported from neighboring countries such as Brazil, Vietnam, Indonesia, and others. Okay. So the global coffee production, according to ICO or the International Coffee Organization, this is actually attributed more to Arabica and Robusta. So Arabica plays a share of 93,423,060 kilogram bags, while Robusta shared 70,211,060 kilogram bags. So minor production is attributed to Coffea liberica that are produced by few countries like first, the Philippines, Malaysia, Indonesia, and Uganda. Okay, so this morning, I know this interests a lot of you. And did you know that since the publication of Davis about the high extinction risks for coffee due to changing climate sometime in 2018, this led to the investigation of potential or other coffee species. For example, according to Davis, these 9 species of coffee are actually cultivated species. And Arabica, Eugenioides, Stenophylla, or the Sierra Leone coffee, had a capping score of 80%. So this is very comparable to a specialty coffee or fine Robusta. Okay? So, oops, parang nag-hang. Nakita po ba? Okay. So, examples of this other species of coffee with economic importance are the Coffea brevipes, the Coffea congensis, and Coffea stenophylla. And their capping scores were investigated by Davis and Bertrand. So this plant of Coffea brevipes, okay, and the matured cherries, yung nasa baba. And then for this one, it's the Coffea congensis. And yeah, these are the ripe cherries of the congensis. This is Stenophylla. Okay. So this is a ripe fruit or cherries of Coffea stenophylla. Okay. So the figure on the right shows the beans, the sizes of the brevipes, congensis, and stenophylla. So the figure on the right shows yung sensory profile of the 3 species of coffee. So as you can see, they possess sensory profiles more of the Arabica than Robusta, which implies that when processed properly, they would produce a good cup quality like that of Arabica. Okay. So, among the 3 species investigated, Coffea stenophylla had the most similar sensory profile as that of Arabica. It's not Arabica, but Arabica-like in sensory characteristics. So it possesses cupping notes common to Arabica such as yung floral, yung berry, yung fruity, at saka citrus flavor notes. So actually, the tree at my back is Coffea stenophylla. This is located in the germplasm collection of the Central Coffee Research Institute in India. So below is the unripe fruits and leaves of Coffea stenophylla. Okay. So Coffea racemosa is produced in South Africa. So if you happen to join the Philippine Coffee Expo last June no po at the World Trade Center, Coffea racemosa was showcased by one of the speakers, si Charles Denison, and some participants had the opportunity to taste racemosa. So ito po yung racemosa. This is the plant of racemosa, of course, the flower, and the ready to harvest cherries. And on the right is the fruit cherries of racemosa and the what do you call this? The pulp coffee and GCB or green coffee beans of Coffea racemosa. Okay. So, Coffea, ang hirap niyang banggitin. Coffea zanguebarrier, or also known as the Ibo coffee. So this coffee is processed in northern Mozambique. Ano po? It's actually in a particular island known as Corimbas Archipelago. So the figure on the right, no, yung beach was taken in 2019. This part of Mozambique is very hot that you wouldn't expect coffee to thrive. And then few distance away from the beach, no po, uh, there's this coffee drying of Ibo coffee. So the figure on the left is the branch, no, the fruits or cherries and parchment of Ibo coffee. So, dito naman sa baba, it only shows us that Ibo coffee thrive in dry and hot locations. Okay. So what are the flavor notes or sensory profiles of Ibo coffee and racemosa? So racemosa has an aroma of spice, sweet herbs, and licorice. And yung body niya is light to medium with light acidity. Yung flavor notes are blackcurrant, spice wine, spice, cannabis, no po, star anise, licorice, butchu. Butchu is sweet cake-like herb. And then cinnamon, cloves, camphor, violet florals, cereals, and mint. And on the other hand, Ibo coffee has dry aroma of herbal eucalyptus, licorice, and then yung body niya is light to medium with light acidity. And flavor notes are herbal, lavender, jasmine, anise seed, licorice, dark chocolate, and of course in cardamom, eucalyptus, medicinal, vanilla, and also mint. Okay, so with the changing weather patterns, it is likely that this coffee will replace Arabica and Robusta. If you remember yung history ng Robusta, the introduction of Robusta was because of the susceptibility ng Coffea Arabica to leaf rust. That's why people back then resorted to looking for coffee that are resistant to coffee leaf rust. And then eventually, ito nga pong Robusta at saka Liberica came into the scenario. And then, we needed to actually look for species that are resilient to drought, that are, what I call this, resistant to pests, emerging pests and diseases, and other abiotic and biotic stresses. So although these species no po, are not available in our country, the need to conserve no po, the genetic resources of coffee in our country is imperative. So I would like to introduce Cavite State University, specifically ano po, the National Coffee Research, Development and Extension Center. So CVSU, through the NCR deck, serves as the national repository for the genetic resources of coffee in the Philippines. So these are maintained as XC2 in a field gene bank where there are 27 Arabica accessions, and we found that 5 were duplicates. Then there were 29 Robusta accessions, around 150 excel sa accessions, and around 36 to 40 Coffea liberica or liberica accessions. Now, yeah, the gene bank gave us the opportunity to identify and characterize the coffee in the Philippines. And we use morphological as well as DNA-based markers to study them. And morphological markers, I think these were mentioned last time by Dr. Cao. Ano po, these are visible traits or phenotypes of the organism, and they can be used in the assessment of plant that serve as basis for at least no po in the context of varietal registration. They can be used to determine if a particular accession is distinct, if it's unique or stable, or yung tinatawag nating DOS system. And on the other hand, yung mga DNA based molecular markers, actually these are sequences of DNA that are specific to the sample, and they are readily detected. So DNA-based markers are found in specific location in the genome, ano po, and they can be used to flag the location of a particular gene or the inheritance of a particular trait. Okay. So ano ba yung mga morphological markers? For our farmers, ano po, hindi po itong mga nakakatakot na terms. But actually, morphological markers are just this one, at least in coffee. Ano po, the overall appearance ng ating coffee plant, yung cherries or yung fruits, ano po, the flowers. And oftentimes, these morphological markers are nakalagay siya sa isang descriptors. For example, the figure on the right, the descriptors for coffee. And it provides an international format and para sa ganun, mas naiintindihan ng nakakarami. So information includes such as yung characteristics ng plants, yung ancestry ng plants, yung collection data natin, yung characterization parameters, and also yung evaluation parameters and others. Okay. So ano ba yung mga advantages ng morphological markers over that of the DNA-based markers? Of course, they are readily available and they serve as the most direct measurement ng phenotype natin. So, in using morphological markers, we don't need a sophisticated equipment. In fact, ruler lang or caliper is okay na or pwede nang mag-conduct ng morphological assessment. However, the disadvantages of this marker is that they are limited in number. And oftentimes, they are affected by the environment. For example, when you grow one plant or one variety in this location, it changes. Some of the traits would change in different location. Okay. Mababa din yung polymorphism ng morphological markers, and they are only available at a particular stage. For example, we would like to examine the fruit color. So, saka lang natin ma-examine yung fruit color when the plant or the coffee plant is in the fruiting stage. And we wanted to look at the cupping profiles ng isang coffee. So that would only be available when nakapag-harvest na tayo ng coffee. Okay? Ano naman yung advantages ng molecular markers over the morphological markers? Of course, yung mga molecular markers are numerous and abundant in the genome. Tapos, hindi sila naapektuhan ng environment. So if we plant this, if we use a variety of coffee in Cavite, so we would be getting the same result when these plants are taken from, for example, in Benguet. So they are not affected by the stage of the plant. Okay. So we conducted a study on the morphological characterization of coffee. Actually, this research was initiated due to a particular government agency or organization. Organizations were reaching to us asking for technical assistance on how to properly identify coffee. Kasi napaka-importante that coffee is properly identified, at least to this organization, because kung hindi tama yung pagka-identify ng coffee, then yung ating minamass propagate na coffee ay hindi tama. Okay. So, There were also farmers na tumatawag sa atin, nagpapatulong, paano po ba ma-identify itong coffee na 'to? And in FB 'no, marami ring post na, sasabihin ito ay robusta coffee, pero if you are to look at the traits na distinct, yun pala ay, trait ng isang Arabica coffee. So we use 20 accessions no po or varieties representing the 4 types of coffee. So gumamit po kami ng leaves, ng flowers, ng fruits, and the whole plant to characterize the accessions using 23 morphological traits. So 18 were quantitative traits and 5 were qualitative traits. So the results of this study were analyzed using NTC software version 2. And we also validated some of our results in the 197 accessions of coffee at NCR Deck Germplasm. Aside from that, we reached out to scientists who had previous publications on the characterization and identification ng coffee. And yung results namin did not revolve on the diversity of the traits only. So we thought of developing an identification key that can be used by farmers, by plant material inspectors, and even researchers. Okay. So, as for the results of this study, we found out that napakataas ng genetic diversity ng leaves, ng fruits, at saka ng flowers. Overall, yung genetic diversity is or range from 0.5 to 0.99 except for flower color. Okay? Ayan. So, yung study namin did not just list or show the various traits for a particular descriptor. For example, tumigil. Okay, so one of the traits, important trait that can be used to differentiate yung 4 commercially cultivated species is the overall leaf orientation. So mamaya po siguro after ng talk, puntahan nyo yung kape, tanim na kape sa likod ng bahay nyo. And then ano kaya yung overall leaf orientation ng aking kape? So if they are drooping or downward yung kanilang mga leaves, then this can either be Arabica or Robusta. But if the leaves are upright, I should say, so then this is either Excelsa or Liberica. And another important traits that we determine are the leaf margin, yung leaf size and texture, at saka yung leaf index. So yung leaf margin, okay, so kung mapapansin natin dito sa figure, Arabica and Robusta ay merong wavy or yung technical term for this is undulate yung kanilang leaf margin. On the other hand, yung Liberica, and Excelsa ay non-wavy yung kanilang leaf margins or tinatawag na entire. Okay? So this leaf margin is very important to determine if this is Arabica / Robusta or Liberica / Excelsa. And on the size of the leaves and the texture of the leaves yung Robusta napakalaki, malaki yung leaves ng Robusta compared to that of Arabica. However, yung Robusta ay non-leathery, medyo manipis siya compared to that of Arabica. On the other hand, yung leaf index can be used to identify Liberica and Excelsa. So yung leaf index tells us the narrowness of the leaves. Yung Liberica is narrow, yung kanyang leaves, while the leaves of Excelsa ay mas broad. So kitang-kita na malapad yung dahon ng Excelsa compared to that of Liberica. Okay. So another important traits are those of the flowers and the fruits. So yung number of inflorescence is a highly discriminating trait in coffee. So yung number of inflorescence would also tell us the clustering of fruits along the nodes ng coffee. So the number of inflorescence of Arabica, Robusta, and Excelsa is remarkably high. That's why yung mga nodes, yung mga fruits ng Excelsa arabica and robusta are highly clustered. And on the other hand, yung clustering ni Liberica ay yung number of inflorescence ni Liberica ay kakaunti lang siya. That's why the fruits of Liberica appear singly along the branches. So this is one of the most discriminating trait to identify also Liberica from Excelsa. And another one is on the size of the fruits. Arabica, Excelsa, and Robusta have sizes of fruits or cherries na maliit. However, yung Liberica talagang napakalaki yung kanyang fruits or yung kanyang cherries. In fact, we were able to encounter a Liberica accession na kung saan mas malaki pa siya kesa sa old 5 peso coin, yung bronze na 5 peso coin. So this trait is very important to identify Liberica from the rest of or from the 3 types of coffee. Okay. So actually, most of the difficulties ng ating mga farmers or ng ating even researchers is on identifying Excelsa from Liberica. So in fact, even yung na-mention ko kanina, even yung mga government employees who should be capacitated to identify Excelsa from Liberica ay medyo nalilito pa din. So this is not surprising kasi si Excelsa and Liberica really belong to the same species. Katulad ng sabi ko kanina dun sa taxonomic classification, Coffea liberica variety de Weuvray is the Excelsa. It's also known as Coffea de Weuvray. Or in Coffea liberica variety liberica is the Liberica. However, although they belong to the same species, what is clear is that these two are or can be Excelsa and Liberica are distinct from each other. So, ano ba yung mga traits that we can use to identify or to differentiate Liberica from Excelsa? Number one is the number of inflorescence. So this trait was actually the most discriminating trait found by Indiahe 2005. So mababa ang number of inflorescence ni Liberica while Excelsa has a very high number of inflorescence, making the fruits highly clustered on the branches. And in contrast, since hindi clustered yung inflorescence ni Liberica, then they appear singly along the branches of the coffee plant. And then other one is the fruit size or yung fruit volume. Napakalaki po talaga ng bunga or fruits ng Liberica, while Excelsa is mas maliit po talaga siya compared to that of Liberica. Yung shape index ng Liberica very narrow, while Excelsa has a very broad leaf index. Yung leaf orientation, as I mentioned, is that both are upright and yung leaf margins ay both have non-wavy or entire leaf margins. So we tried looking at the separation nitong 4 na types ng coffee, and indeed as you can see in this figure, they are separated from each other. For example, the cluster 1 is the Arabica cluster. Then yung cluster 2 are the accessions that belong to the Coffea canephora. Cluster 3, meron lamang pong 2, but these are Liberica accessions. Cluster 4 are also Liberica accessions. And the last cluster is yung Excelsa accessions. Since it has been difficult for others to identify yung Liberica from Excelsa, we also increased the number of accessions using those 3 highly discriminating traits po. So we use 12 Liberica accessions and 15 Excelsa accessions. And our cluster analysis tells us that the two coffee types were discriminated by fruit size, by the number of inflorescence, no po, and also the leaf index that tell us the narrowness of the leaves. So cluster 1 and cluster 3 were Liberica accessions, while cluster 2 and 4 were Excelsa accessions. Okay. Molecular markers were used, of course, to characterize yung coffee accessions that we have in the gene bank. We use DNA barcodes or universal sequences, ano po, and there are other two markers, the inter-simple sequence repeat markers and the SSR markers or simple sequence repeat markers are also known as microsatellites. So the general procedure for the analysis of molecular markers, ano po, includes the gathering of samples from it depends na po dun sa objective natin. Oftentimes, we use the leaves. Then we extract the DNA, and then this DNA are amplified using a machine or the polymerase chain reaction machine. And we subject the products or the amplified DNAs into electrophoresis and eventually document them under the gel documentation system. So for DNA barcoding, we use MATK or Maturase K and ITS. ITS or yung internal transcribe spacer po. So we didn't use the RBCL na ginamit ni na Dr. Cao last time kasi we base actually yung from their results. So hindi naka-discriminate yung RBCL, hindi na-discriminate ng RBCL nila dun sa apat na klase ng coffee. So we used 24 samples of the cultivated coffee, and our results showed that ITS did not separate Arabica from Canephora, no po? And Coffea liberica, that includes the Liberica and Excelsa, form a separate clade but did not separate Liberica from Excelsa. Okay. On the other hand, yung MATK, ano po, all three species separated from each other. So this is the same results na na-observe ano po in that yung Liberica cannot be separated from Excelsa. So the result simply implies that universal markers such as ito nga po, yung MATK at saka ITS are useful in identifying yung species ng coffee. The only drawback of this is that they cannot be identified. I mean, the taxons lower than species level cannot be identified. And then, of course, we use ISSR markers or the inter-SSR markers. So we analyze the present or absent bonds and bonding patterns associated with the species or yung type or variety ng coffee. For example, using the primer UBC 841, we can see that Robusta did not show any bond. This primer, specifically the 200 base pair bond, is diagnostic to Robusta coffee. And on the other hand, ano po, the marker UBC 835, the presence of bond with around 650 base pairs indicates that the sample is a Liberica coffee. Okay, we summarize the 7 primers, only the reproducible 7 primers ng inter-SSR to identify markers and bonds and also yung bonding patterns that are diagnostic to coffee, specifically on the type of coffee and the varieties of coffee. For example, those bonds highlighted with orange, no po, are diagnostic to Coffea liberica. So those in red are diagnostic to Excelsa. Blue and green are diagnostic to Arabica and Robusta respectively. So this sample, yung number 12, okay, ito po, sample number 12 is known in the center na Arabusta because of the combined traits ng Arabica at saka ng Robusta. So the result of this study invalidated yung claim namin nung una that this variety is a hybrid of Arabica and Robusta. Rather, the Arabusta that we are talking about is just a Robusta variety. Okay. Several bands were found to be diagnostic to a specific variety of coffee. For example, this band highlighted in green, when absent using the marker UBC 841, means that the variety is FRT 11. So FRT 11 is an NSIC registered variety. So we can actually use this band to identify that this is an FRT 11 variety. So this band in purple color is diagnostics to CVSU NSIC Robusta. Actually, yung CVSU NSIC Robusta is the only NSIC registered Robusta variety other than Nestle. So this one in orange is diagnostic to CVSU NSIC Liberica, the only Liberica registered variety in the country. And yung blue naman is diagnostic to Red Catura. So sa mga varieties that do not have a specific diagnostic band, the unique banding patterns na po is considered. For example, yung variety na Granicai standard, it is expected that it should generate this banding patterns when run using primers UBC ito 826, then UBC 834 at saka UBC 836. Ayan. So overall, these are the diagnostic markers no po, that we use for the different types of coffee as well as the different varieties of coffee. And for proprietary reasons no po, we didn't specify the specific band diagnostic to each variety. Ayan. So we also use SSR markers to characterize our coffee accessions. These three markers, yung SSR, CBS, C, D at saka X were found to be diagnostic to the three species ng coffee. So this accession box in green are Arabica accessions. This one are Liberica or Excelsa, and these are Robusta accessions. So for marker SSR CVSC, we expect this band in Arabica, this band in Excelsa, and Excelsa or Liberica, and this band for Robusta. For marker D, so these are the expected bands for Arabica, and these are the expected bands for Liberica or Excelsa, and ito naman po sa Robusta. For the last marker, okay, yung X, these two are the expected bands ano po, for Arabica, and these are the bands that we expect for Liberica and Excelsa, and this band is the expected band for Robusta. Okay. So we also analyze the genetic diversity of our accessions no po, using SSR markers. And overall, the genetic diversity is from 0 to 0.73. So the genetic diversity of Arabica within Arabica accessions is 0 to 0.36. In Robusta, it's 0.09 to 0.2. So I'll tell you bakit kaya masyadong mababa yung sa Robusta. And in Excelsa, slash Liberica, it's 0.11 to 0.73. So yung sa Robusta, napakababa po nang nakita po naming diversity because most of the accessions we gathered were NSIC varieties or came from NSIC varieties. So yung mga FRT varieties ng Nestle. So the SSR markers used resolve the separation of the three species. However, again, Liberica could not be distinguished from Excelsa. So ito po. So as you can see, may mga Liberica accessions. Liberica accessions sa taas and then and then merong interclustered yung Excelsa and so on. So we really cannot differentiate yung Excelsa and Liberica. And I will talk more about this one in a while. Okay. I would like also to emphasize that we're using SSR markers to determine the authenticity of GCB as well as roasted and ground coffee sold in the market. So we optimize the extraction of DNA in GCB as well as ng RCB at saka ng ground coffee. For the green coffee beans, medyo mas madaling extract ang DNA. However, in roasted coffee, well, as expected, since it's already roasted, medyo mahirap extract yung DNA coming from roasted beans as well as those that are ground coffee. So, different sets of markers were used. For example, yung marker R278, the presence or absence of this bond indicate Liberica or Excelsa. So, Liberica 3, Liberica 5, Excelsa 1, Excelsa 3, Excelsa 5, and the blend ng Excelsa Robusta Arabica or the IRA 1, and the other blend consisting the 4 types of coffee or the Real 1, okay, they are expected to contain or to have this bond. And so we sampled GCB sold in the market, but of course to protect the name of the seller or the identity of the seller, we repack our samples in uniform plastic bags para hindi masyadong ma-identify uh, yung mga coffee na yon. And I would like to show you one sample that we had. For example, this lot or ito binili namin in region 4A. And yung label niya talaga, ang ganda-ganda pa yung ng label niya. It's pure Robusta. So it's GCB sold in region 4. And we code it as G3 in the gel. Okay? The first thing we did is to test its purity using yung SSR primer 123909. So Robusta does not have this bond. Okay? Then we check the contamination. And as you can see, this bond is specific for Liberica or Excelsa is present in the sample. Thus, we ascertain that this sample is not true to its label. Okay? Ayan. I hope you have seen this advertisement na po kahapon ni Jollibee. And syempre, sabi ni Jollibee, 100% Arabica beans. Jollibee ha, umaasa kami sa'yo. Okay. So, kanina nabanggit ko po about the differentiation or non-differentiation of Liberica from Excelsa. Ano po? And we use a larger set of Liberica and Excelsa samples to confirm if Liberica can be differentiated from Excelsa. So yung Excelsa is colored red, while yung Liberica is colored blue. We use 73 samples in all, tag to 35 ng Liberica and 38 na Excelsa. So the yellow and orange are the control, yung Arabica and Robusta respectively. So we use the same sets of markers, the 16 SSR markers that generate 68 alleles. And there are 3 major clusters formed. The first cluster consists a combination of Liberica and Excelsa. The second cluster has more number of Liberica, but considerable number of Excelsa accessions clustered with it. Cluster 3 consists of Liberica accessions, but there was one accession that joined them. So as you can see from this dendrogram, there was no clear separation between Liberica and Excelsa. So medyo sabi namin, why not increase the number of samples? For example, we included 20 more samples that are a combination ng Liberica and Excelsa. So we code it as LE, LE to show that these accessions are more of the Liberica types. And from our results, there were 3 clusters formed. The first cluster consists of a combination ng Liberica and Excelsa. So yung second cluster naman consists of Liberica, Excelsa, and LE. While cluster 3 consists of Liberica accessions, but there were two LE accessions that joined them. Again, with this very high number of accessions, it showed us that Liberica cannot be separated from Excelsa. So yung result ng aming study is actually in contrast with those that were found out by Indiahe in 2005. No po? Another researcher was that of Haymon. So sa kanilang study using rDNA, they were able to separate Liberica and Excelsa. And also that of Maureen in 2007, wherein they used nuclear and plastid DNA sequences. They were able to separate Liberica and Excelsa. So ano kaya yung possible reason why this happened, at least to the Liberica and Excelsa of the Philippines? So ano yung implication nito to our seed system, to our what do you call this, conservation efforts in the Philippines? So if you remember, coffee was brought to the Philippines sometimes in 1700s. Ano po? Although I'm not so familiar, or I do not know much about the history of coffee in the Philippines, and I believe marami pa rin i-correct doon sa history ng coffee in the Philippines. I assume that what was brought to our country were seeds and not seedlings. So we know that Liberica is a highly cross-pollinated crop. And the most valid reason for the non-separation of these two is the use of seeds in the propagation of Liberica in the Philippines. So since its introduction in the 1700, no record show us that Liberica was propagated through clonal means. So the crop's high degree of cross-pollination resulted in trees that are now mixtures ng Excelsa at saka ng Liberica. So actually, we can liken this to the history of Filipinos, no? The original Filipinos were short, black, and with flat noses. Due to marriages with other races as the Spaniards, Chinese, Japanese, Americans, and others, the modern Filipinos are already a mixture of different races. However, nandun pa rin, very distinct pa rin yung Filipino traits. For example, we still have the flat or yung semi-flat na nose. Basta hindi pointed yung nose natin. The color of our skin is brown, indicating that black was mixed with white. At yun na pala yung last slide. Okay, so I think that's all. Medyo nag-exceed po ako sa oras, but of course, thank you so much po. And if you have questions, you may contact me through this email addresses po. Thank you and good morning po sa inyong lahat.
Thank you very much, Dr. Miriam Baltasar, for that informative, comprehensive, and quite technical presentation po. So I know eager na po magtanong yung ating mga audience. So now to address the audience question, let us proceed with the open forum. To serve as our moderator for this episode, let's welcome Ms. Sheni A. Hilado from the La Granja Research and Training Station. Miss Sheni, the virtual floor is yours now po.
Thanks, Jessa. Hello, good morning to all attendees via YouTube, Facebook, and Zoom platforms. Good morning po, Dr. Baltasar. So para po sa mga kakapasok, yes po. Para po sa mga kakapasok o kakabalik lang, you are currently attending the Coffee Webinar Series, Episode 3, Identification and Genetic Characterization of Philippine Coffee, presented by Dr. Miriam Baltasar. So kakatapos lamang po i-present ni Dr. Baltasar ang the taxonomy of coffee, where does coffee grows predominantly, dun po sa diniscuss niya yung coffee belt na tinatawag, yung other coffee species with economic importance and potential. And Dr. Baltasar also discussed about conservation of Philippine coffee, identification and characterization of coffee, yung mga markers po, yung morphological at molecular identification and characterization using traits, tapos yung mga molecular markers, and there. And so I believe this is the time po to ask or clarify information from Dr. Baltazar. So para po sa may mga katanungan sa ating mga Zoom participants, please use the Q&A chat box or raise your hand to be recognized. Para naman po sa Facebook and YouTube viewers, please use the comment section and wait po natin na i-acknowledge them. Okay, so we have... ready ka na po, Doc?
Yes po. Opo.
Sige po. So for our first question from Sir Bernard Panabang. So hello po ma'am, what type of coffee do you prefer for your drink?
It's Arabica. Pero gusto ko yung nahahaluan ng Excelsa or Liberica. Kasi gusto ko po talaga yung matapang na coffee. Medyo hindi masyadong matapang kasi yung Arabica. But of course, I like the floral notes ng Arabica. Ayun po.
Okay na po ba, sir? Okay, so thank you po ma'am. So for the next question po, from Sir Joel Don Untalan from the Philippine Genome Center, Mindanao. What substitution model did you use in constructing your phylogenetic tree? Thank you.
Sir, what do you mean po na substitution model? Is that on the ITS or MATK or doon po sa SSR saka yung ISSR po?
Sir Joel? Meron po siyang follow-up question, ma'am. From Sir Joel po, ang sabi niya po ay, are you also looking for other omic approaches in characterization of different varieties of coffee?
Opo, actually, not only genomics po, but I just would like to emphasize din ano po, na aside from SSR markers, we recently po kasi may na-approve po kaming project no po, under the DSTP card, and we'd be looking at the SNPs no po. The only problem is that wala po tayong reference genome for Liberica. We have for Excelsa and Robusta, meron na po tayong reference genome for this. But for Robusta kasi it's not publicly available. It's only for Arabica. But we know Arabica is tetraploid. So I think medyo mahirapan kami. But of course, one of the prospects is the future prospects is that hopefully we can also collaborate with agencies such as the Philippine Genome Center is for the sequencing po ng ating Liberica, specifically po ng CVSU and SIC Liberica. And also, there are, of course, metabolomics approaches being conducted for to identify yung ating coffee. And we also have one scholar, currently studying po sa Australia. She's doing lipidomics on coffee to identify yung ating mga varieties po ng coffee.
Okay, thank you po ma'am. Doon po sa tanong kanina ni Sir Joel, ang sabi po niya ay nucleotide substitution model po from the SSR markers.
Are you referring po ba doon sa specific analysis, sir? We did not use substitution model. So actually, we just scored yung ating bonds. We have the expected bonds in SSR. However, merong mga bonds that consistently appear. We actually repeated yung PCR run for 3 times. And yung consistent bonds, even if they were not declared as the expected bonds, ginamit pa rin po namin yon.
Okay, thank you po ma'am. I hope Dr. Baltasar answered your question, Sir Joel. Okay, for our next question, from Sir Bernard Panabang, would it be fair to say that somehow the Philippines has become a sub-center of genetic diversity for Liberica excelsa coffee? What are its implications in terms of plant breeding, especially for resistant against pests and diseases?
I cannot say na yes kasi we didn't have extensive study on the characterization of Liberica and Excelsa. So what I mean to say is that we haven't been into the detailed characterization ng ating Excelsa and Liberica. However, ano po, we have seen diverse collections in the gene bank, no po. But I cannot say na ito po masabi nating subcenter ang Philippines ng Liberica and Excelsa. Probably not. Doon pa rin po sa sa Africa. Nandun pa rin po yung Center of Diversity.
Okay, thank you po, ma'am. And another one po from Sir Bernard Panabang. Oh, okay, it's done na po pala. Okay, from Mr. Speed Crisostomo from UPLB po, is there a crop improvement or breeding effort being done by Cavite State University? If yes, what are the target rates? If there is none, is it possible to collaborate with CAFS for this initiative?
If we talk about the formal breeding programs or activities, wala pa po. In fact, matagal na akong sinasabi na why not do this and do that. Pero sabi ko magre-retire na ako, wala pa akong napoproduce na ano. However, actually nakita na namin yung need kasi po it's not easy for us to access germplasm for other countries kasi nga well, coffee is restricted commodity. Hindi po talaga siya mutually shared among countries. So talagang napakalaki yung role natin in in developing new varieties na po that are that address mga problems, especially problems in the future such as pests and disease susceptibility, abiotic and other biotic susceptibility and others ano po. Formal breeding, wala pa po. Informal, yes, because we've been doing, alam mo yung trial lang ng pollination. But actually, we have seen the onset of anticis, yung plant breeder naman si Sir, yung anticis ng coffee that is closely related. And actually, it invalidates yung common number, I mean, yung schedule ng anticis ng isang plant. 'Di ba mas ina-expect natin yung anticis or yung pagbo-bloom ng flower is or sorry, yung peak ng meiosis or gamete formation is between 10 to 1 PM. However, in coffee, it's ibang-iba siya. Actually, madaling araw po siya and it requires around 48 to 72 hours after such amount of rainfall. So yun po yung naging medyo ano sa amin, naging hadlang para isa sa mga naging hadlang kasi binabantayan namin yung anticis but it has been very difficult for us to do that. Informally, yes po sir. Formally, wala po.
Yun. So I hope po na magkaroon po ng collaboration through that question no ni Sir Speedy. Okay. So thank you po ma'am. So from Mr. Ray Bacalangco po from La Granja Research and Training Station. His question po is for farmers, how can they identify the coffee type while it is on seedling age or stage or before flowering stage? At what exact age or stage can the farmer identify their seedlings? Baka hindi nila maintindihan ang genetic markers.
Yes. Actually, one of the drawbacks talaga nung ating study is that you cannot identify yung ating mga types of coffee at the seedling stage. And actually, this is very important kasi po, napakalaki po ng seedling demand no po ng coffee in the Philippines. Katulad po in the next 2 to 3 years, ano po, malaki po talaga ang ating purchase ng coffee seedlings. However, unfortunately, wala po kaming na-identify na morphological trait that could distinguish these 4 types of coffee. We have one thesis student who's working on this, pero hindi pa rin po siya tapos, kaya wala pa rin po akong masasabi today the trait that could distinguish the 4 types of coffee at the seedling level.
Okay po. Sir Ray, I hope Dr. Baltasar answered your question. So meron po tayo dito ma'am from Miss Melissa Makasaet. How much would it cost to have coffee plants analyzed to determine the types or cultivars?
Sir, are you referring to the DNA base?
Ma'am Melissa, Dr. Baltasar is asking po if you're referring to molecular po ba na pag-analyze? Maybe po ma'am, pag molecular po, how much does it cost?
Apo. Actually po, we don't do molecular DNA-based identification right away. So i-apply po muna natin yung morphological characterization. 99% mare-resolve na using the morphological characterization ng coffee. Kung hindi na po talaga kaya, then we'll be doing the barcoding. Barcoding cannot identify the Liberica and Excelsa. Wala po kami exact price na po kasi ay napakahirap po sa amin right now na po. And also wala pa po kaming established na price rate. So habang wala pa po, hopefully it's still free na po. Wala pa po talaga.
So, ayun po. Ma'am Melissa, free pa po sa ngayon. And from Ma'am Kat Luzon, good morning. First of all, congratulations on a very rigorously conducted study, Dr. Baltasar. I would just like to clarify the part of the presentation where you were showing how Excelsa and Liberica could not be clearly resolved. Might I clarify again what is the history of Liberica and Excelsa? Are they hybrids of other species, or are they species in the process of completely separating from each other? Process of introgression.
Actually, the Excelsa and Liberica are distinct groups of the Coffea liberica. Hindi po siya hybrid. Hybrid in the context of plant breeding, ano po, kasi ang hybrid po kasi ay cross ng two distant parents, and then yung kanyang offspring ay merong mas mataas na kalidad kesa sa dalawang parents. And hybrids cannot be propagated via seeds kasi yung mga traits niya magsesegregate. So Excelsa and Liberica are not introgress, but they are of the same species. It's just that they are distinct two groups ng Coffea liberica. Ayun
Okay
po.
po. Sige po. Ma'am, meron po tayong ulit question from Sir Bernard Panabang. So, are you also looking to importing foreign germplasms of coffee for use in plant breeding?
Yes and no and yes. Opo. Actually, yun nga, meron kaming initial conversation with CRRI, the Coffee Research Institute in India. And they have these collections, yung most of the wild species ng coffee that I mentioned, yung Racemosa, yung Ibo coffee meron din sila, at saka yung Stenophylla. However, as mentioned, ano po, para lang po sa kaalaman po ng marami, yung coffee po kasi and some other political crops like cacao, they are not listed in the Appendix 1 ng FAO, Food and Agriculture Organization, na kung saan nakalagay doon na hindi pwedeng mutually shared yung mga genetic resources ng coffee, no po. So there's a need for bilateral agreement between countries in order to access this collections. Unlike rice, ano po, na madali lamang po tayong makahingi, for example, sa IRI, or other countries can easily request accessions, for example, sa IRI. Kasi rice is mutually shared among countries. But coffee, hindi po talaga siya available for exchange between countries. So we need bilateral agreement about exchange of materials. But syempre, we desire, of course, to have this species of coffee kasi it would increase yung diversity ng coffee we have in the Philippines. Hopefully, magkaroon po ng linaw yung ating pong initial communication with CRRI. Thank you.
Okay, thank you po ulit, ma'am. So from Sir Joel Don Untala naman po, have you also looked into consideration in characterization of these varieties in terms of identifying the specific genes associated with desirable traits such as flavor, aroma, and disease resistance? Thank you.
Yeah. Actually, yung group ni Dr. Cao, ano po, mentioned about, I mean, they are doing this one. However, siguro to complement yung kanilang projects on identification of markers associated with a particular important trait, there's a need to confirm yung expression ng trait sa field. So, we are actually talking with each other to really do the screening, for example, doon sa germplasm collection po ng cough.
Okay po. Another one po from Sir Bernard Panabang. Do you sell or share certified coffee clones in your institution? Something that can be bought online? Because it is difficult for starting farmers who do not have transportation means to buy coffee seedlings.
So I suggest that pumunta po kayo sa malapit na accredited nursery grower na malapit po sa inyo. So I think in CVSU sa NCR Deck ngayon, napakarami po nang nag-order ng NSIC Liberica. So hindi ko alam yung status ng order, but parang naririnig ko na medyo may kakulangan po sila sa mga seeds na ipo-provide or ibebenta po this coming season. So pumunta po kayo, sir, magpatulong po kayo sa Department of Agriculture na malapit po sa inyo so that they can identify or refer yung accredited nursery owners po.
Ayan po. Thank you po, ma'am. Para po kay Sir Junicio Felicen, siguro po yun na din po yung sagot ni ma'am kasi nagtanong po siya kung saan po makaka-avail ng seedlings po ng coffee. So, ayun po. Meron pa po bang gustong magtanong? Please send po your questions sa ating Q&A chat box. Okay, so we have here. Meron po tayong wait lang po. So, Sir Maximo Gahulin. Hello, sir. Sir Maximo, you can ask your question po. Okay, mukhang naka-mute po si Sir Maximo. Okay, so let's have na lang po muna Sir Emilio Climaco. Sir Emilio, you can ask your question na po.
Can you hear me?
Yes po.
All right. Hello, Dr. Yam. I'm Emil from Shepherd's Farm. I actually got interested when I saw the ad ng UPLB about coffee. I'm not into coffee, but I am the president of our organization in CPAG. And we were given, at nasa akin ngayon sa farm ko, yung 5,000 seedlings of this coffee type. At hindi ko alam kung anong variety siya. So I was a little bit problematic kasi when people ask me kung anong variety siya, hindi ko alam, hindi ko masagot. That's the reason why I attended this seminar. And I was listening intently to you and I found out na Liberica yun based on the morphological characteristics. So tama yung hula ko when people were asking me kasi pinapamigay ko eh. Unfortunately, ay meron pa ako kasi kunti lang. I'm from Pagsangan, Laguna, by the way. So, unfortunately, kunti lang sa amin yung kumuha ng coffee seedlings na ito because of the historical, what do you call this? Yung distribution ng seedlings na may mga nagtanim pero hindi nag-prosper. So that's one statement that I'd like to say. So I'm just perplexed na binigyan kami ng DA, ako naman, without doing due diligence, that was my fault, binigyan kami ng DA ng 5,000 seedlings ng Liberica coffee without asking kung ano yung need namin. So, partly fault ko yung kasi tinanggap ko. Pero since andito na to, Ang concern ko, which I'd like to ask you now, I know this is a bit far from what you discussed, pero ang question ko is, given na meron na kaming Liberica coffee, based on your understanding and your maybe research, if you did, dito ba sa Pagsanghan, Laguna, or any part of 4th District or 3rd District, ay pwede mag-thrive ang Liberica coffee?
Mhm. Okay, sir. Sir, ang isa kong katanungan is, ano po yung elevation ng area?
I don't know particularly, pero just to give, ka-level kami ng siguro a little bit higher than the Laguna de Bay. So, knowing kung Laguna de Bay ang ating reference, ay within the Laguna area, Doc. So, just
Apo, sir. So actually, in terms of elevation naman, kasi oftentimes, ang Liberica, Excelsa at saka Robusta, they grow in lower elevation. Pero ang Arabica po, they really require high elevation para maging productive po sila. So if yung mga seedlings na yon, opo, pwede po siya sa elevation na ganyan, sa pagsanghan. But of course, it's necessary for you to assess yung site, ano po. And ang NCR, sorry, since nabanggit niyo po kanina, you are a member of a cooperative, then actually ito po yung gustong i-capacitate or tulungan po ng NCR-DEC yung mga cooperatives. So you can request site assessment po sa National Coffee Development Extension Center para po ma-assess kung suitable po yung ating area for coffee production.
thank you, Doc Yam. Yan po ba ay nasa VSU, yung Development Center?
Cavite State University, sir.
Yeah, Cavite, sorry, Cavite. So, nasa Cavite po siya. Okay, so to whom are we going to address the letter if we'd like to request for assessment?
So, idiretso niyo po sa president po namin and then ibababa naman po niya sa concerned unit which is the NCR-DEC.
Okay, okay po, Doc. And
However, sir,
lastly,
gaano na po kalaki yung mga seedlings? Baka kasi nag-J-root na sila. Matanda na ba yung mga seedlings natin?
yes, Doc. Actually, that's what I was about to ask you next. Na given na medyo matagal na, it was distributed to us, let me just trace back. Today, November. Ano po, binigay po ito sa amin mga August. Tapos maliliit po siya. Ngayon pa siguro mga ano na siya, half a meter. Half a meter na yung height niya.
Kaya ko po tinatanong, sir, kung medyo malalaki na talaga yung seedlings natin kasi baka kasi naka-develop na siya yung primary root po ng seedlings nag-J na siya. If that's the case na po, pag tinanim natin siya sa field, hindi na po yun magiging productive.
Oh.
So, iwasan po natin magtanim po ng J-rooted seedlings kasi malulugi lang, sir, tayo kasi hindi siya magiging productive in the end.
Oh, good thing you told me that, Doc, kasi what I'm planning to do is ipamigay ko ito sa mga friends and even I'm a part-time teacher kasi ibibigay ko ito sa aking mga students. So, ano pong gagawin ko para ma-check kung J-rooted na sila or hindi? I have to
Okay,
open?
so mag-sample kayo. Opo, mag-sample po kayo and then tignan niyo po kung yung primary root po ng seedling ay
Hello?
medyo, kaya siya tinawag na J-root kasi
Kailangan,
medyo nag-J na siya. Oo. Kasi pagka ganun, sir,
so,
eventually hindi makaka-penetrate yung ating root pababa, diretso. So, hindi na magiging stable yung ating plant kapag tumanda siya. And as a result, hindi na siya magiging productive.
okay, Doc, medyo nawala yung first part, hindi ko narinig. I don't know kung nasagot niyo yun. So, ang gagawin ko, Doc, ay bubuksan ko yung plastic ng seedling para makita ko kung nag-J-root siya tama. So,
Yes po.
okay po. So, with that, pag nakita ko na nag-J-root na nga, hindi ko na lang po ipamigay. Tama po ba?
Opo, opo, kasi wala na eh. Hindi na yun magiging productive, sir. Yun yung isang reason talaga na hindi nagiging productive yung isang coffee plant. Kahit hintayin mo siya, diligan mo siya, lagyan mo siya ng maraming fertilizer, since yung roots niya hindi talaga nakaka-penetrate below, hindi talaga siya magiging productive.
Oh,
Okay.
thank you so much,
If you
Doc.
happen to visit CVSU, sir, you can bring 1 or 2 seedlings para makita po natin.
Yes po. Good thing, Doc, I attended this kasi this December, ipapamigay ko na siya. So, kung hindi ko siya check
Mas
then it's just be magiging waste lang yung aking effort. So, I'm thankful dito sa inyong talk. And for answering my question, yun lang po, Doc and Ma'am Sheena.
Thank you.
Thank you po, Sir Emilio. Okay, balikan po natin si Sir Maximo. Sir Maximo, you can ask your question na po. Okay, mukhang hello, sir.
Hello po, Doc. Good morning
Ayun.
po.
Morning po.
Meron po kami, may farm po na maliit doon sa Oriental Mindoro. Kaya lang po, na-bothered po kami. Wala po gaanong tao na nagtatanim po ng coffee. Pwede po ba kami maka-avail din ng training program para sa pagpa-plant ng coffee? Kasi lahat ng farmers naman po doon ay willing, kaya lang nangyayari sa kanila ay lack of knowledge how to plant coffee. More on, pero yung geographical location naman po ay maganda naman po siya. Marami sa river, pero hindi po sila ganun kanila knowledgeable when it comes of planting of coffee and classification of coffee.
Apo, sir. Sir, I suggest po na lumapit po tayo sa DA Region 4B, si Sir Japheth. I think nandito si Sir Japheth, ano po, para po matilungan po nila kayo. Although ang NCR deck naman po ay very much willing to help you, but I think at least yung proximity issues ay mas masasagot po kasi ng DA Region 4B. So si Sir Japheth po, I hope nakikinig si Sir Japheth. Nandito po ba? Alam ko nandito po siya sa Zoom. Ayun po, sir, mag-reach out po kayo sa regional office ng Department of Agriculture.
Yes po, Doc. Thank you po, Doc.
Okay po.
Thank you po, Sir Maximino. Okay, another one po siguro from, ito po, magtatanong po si Sir Marwin Joseph Magindayaw. Sir Marvin?
Pindot lang.
Sir Marvin? Mukhang nawala na po. Nawala po ata, ma'am. Okay. We have, may time pa naman po, ma'am. So we have 2 questions pa po sa Q&A chat box natin. We have here from Sir John Carlos Ignacio. Are there any SNP genotyping technologies available for coffee?
Hello, sir. Kung sa amin po ay wala po. But I think for PGC sa UP Diliman, I'm not so sure kung meron po sila.
Okay po, ma'am. Another one from Sir Vicente Nake. Thank you very much for having this opportunity. My question is, available na po ba ang inyong nabanggit na coffee sa Mindanao? And if available na daw po yung coffee sa Mindanao po.
Sir, sorry, medyo nakalimutan ko po ata yung coffee sa Mindanao. Alin po kaya doon? Coffee sa...
Yes po, Sir Vicente. If makakapag salita po kayo, you can raise your hand po and I will unmute you. Well, while waiting po kay Sir Vicente, ma'am, meron po tayong question from Sir Joel Untalan po. Can microbiome of the soil and the coffee plant influence the flavor profile?
Yes po, definitely. yung tinatawag po nating terwa. Terwa, opo. Different, I mean, similar plant, similar coffee, when planted in different locations, would give different cap profiles, capping notes as affected by the microbiome. Yes po, sir.
Okay. Meron pa po bang gustong magtanong? Yan. From Sir Junicio Felicen, ma'am, pwedeng makahingi po ng email ulit para kung may tanong ako, maka-message po ako.
Apo, sir. I-type ko po dito sa sa chat box.
Sige po. Thank you po, ma'am. Nag-raise po ulit ng hand si Sir Emilio. Yes po, Sir Emilio.
Hi, Doc Yam. Yes, thank you. Pahabol question, last one. I'm selling coffee actually. I'm reselling coffee sa may pwesto kasi kami sa mall. Ngayon, given na co-op kami at binibigyan kami ng ganitong intervention ng government, may coffee nga na I don't know if we will push through planting this or not. Kunwari po in the future na magtanim kami, tapos most likely siguro Liberica ulit ang ibigay. This is a good variety for us to sell sa aming co-op store. Yun po, Doc.
When you say good, sir, you mean the cup? I mean, yung like,
profitable po, Doc. Like, kunwari, nagtanim kami ng Liberica type ng coffee, tapos namunga na siya, and then process namin. Based on your knowledge, would it be profitable for us na ibenta ito?
sir, may pera talaga sa kape. Talagang i-market, no? Opo, well, of course, may mga factors din naman na nakakaapekto why hindi nagiging profitable yung coffee. Halimbawa, kung hindi po natin pre-process ng maayos yung ating coffee, even if naturally meron siya nung good cup quality profiles, then hindi rin siya mabebenta kasi hindi nga maganda yung pagkaka-process. So marami pong mga factors that could affect yung quality or cup quality po ng ating coffee. So for example po is yung processing methods. Okay, for example, mas maganda ba na gumamit tayo ng fermentation method over the dry method or kung ano man. But of course, please follow good agricultural practices and good manufacturing processes or proper post-harvest and handling activities. Para sa ganun, mabebenta natin yung ating coffee ng mas mataas na presyo.
Thank you, Doc. Follow up po with regard to that. Kasi when we sell our coffee, tinatanong ng customers, the quote-unquote variety. So, I am thinking na customers are after the variety or the type nga, nabanggit nyo kanina, type ng coffee, like Excelsa, Liberica, Robusta. Since we have this Liberica, yung question ko po kanina is, particular ba ang customer sa type ng coffee?
Sir, yes. Ako po personally, hinahanap ko po talaga yung Arabica, pero gusto ko rin kasi talaga ng matapang na coffee. So, gusto ko yung blend ng Arabica at saka ng Liberica/Excelsa para mas matapang yung coffee. Opo,
Yung,
sir. Marami rin
yung
po
po
yung mas
blend,
gusto ang pure lang na Arabica kasi very smooth kasi ang profiles, cupping notes ng ng Arabica.
you mean po ng blend, like pag nag-ground siya, biniblend yung type ng coffee ng Liberica at kunwari 50-50, ganon?
Yes po, sir. May mga ano po kasi, para po mas matuto, sir, kayo about the post-harvest handling and processing po, oftentimes ang NCR DEC nagko-conduct po ng trainings for free po for coffee processing and post-harvest handling. So abangan niyo po, actually ngayon ay harvest season, so probably in the next 2 to 3 months po ay magla-launch po ulit ang NCR DEC ng farm tour as well as training on post-harvest handling and processing.
Okay po, Doc. Thank you so much.
Okay po, welcome po.
Thank you po, Sir Emilio. And siguro po, ma'am, last na po, from Sir Bernard Panabang. Hello po, ma'am. Thank you very much for this very informative talk. What advice can you give coffee enthusiasts like us who would want to venture into coffee farming and breeding?
Coffee farming and
Breeding po.
breeding. Um, sorry sir, yung coffee breeding po ba for for development ng varieties, new varieties po ba? Okay. But anyway, for the coffee farming po, of course, i-follow po natin yung good agricultural practices po on coffee production. And meron din po tayong Philippine National Standards on coffee. Ano po? For example, yung proper handling, proper storage, and then of course, proper post-harvest processing ng mga coffee. So, oftentimes po, kaya hindi masarap talaga or hindi nabebenta yung coffee, it's that hindi po talaga maganda yung pagkakaprocess po niya. But, um, mas maganda po na gumamit po tayo ng, yun nga, i-follow natin ang good agricultural practices. Kasi po, if we know the quality of our coffee, then we can demand for a higher price. So, hindi na lang tayo babaratin ng ating mga customers. Yun, sir. For the breeding, um, hindi ko po masyadong naintindihan yung question, sir, but well, of course, if we wanted to breed new varieties, then we can characterize these varieties, and kung nakita po natin na outstanding po yung kanya pong mga characteristics, then probably we can register that to the National Seed Industry Council, and then eventually para po mas profitable, then we can apply for plant variety protection. Kung yun po yung gustong itanong ni sir.
Yes po, ma'am. Ang sabi po ni Sir Bernard ay yes po, for improving our coffee quality to capture the world market with unique Philippine blends of coffee.
Okay.
Ayun.
So, yun sir, yung processing, of course, it's important to start with a good, a good quality planting material. So, katulad ng sabi ko kanina, J-root, yung pinag-usapan natin with Sir Emilio, ano po. So, do not plant J-rooted seedlings kasi talagang hindi yan magbibigay ng magandang production sa atin. And of course, tignan natin kung yung area natin is really suitable or not for coffee production. Baka naman nagtatanim tayo ng Arabica kasi maganda yung inherently maganda talaga ang cap quality ng Arabica, pero itinanim naman natin sa low elevation. So that will not give us a good cap quality. Good yield, no po. At the same time, habang bumababa kasi yung elevation for Arabica, medyo parang robusta na lang din po siya. So actually, it's because of the high elevation kaya po gumaganda yung cap quality po ng Arabica.
Okay, thank you po, ma'am. Dun po kanina sa question pala ni Sir Vicente, ang ibig niya pong sabihin ay kung available na daw po sa Mindanao yung Liberica Excelsa po. Yun daw po yung tinutukoy niya.
Okay po. Sir, hindi ako familiar kung sino po yung accredited nurseries sa Mindanao on Liberica and Excelsa. But if the question is kung nasa Mindanao, may mga tanim ba na Liberica and Excelsa sa Mindanao? Napakadami po. In particular, sa Sultan Kudarat, marami pong Excelsa and Liberica po doon. But for the seedlings, lumapit po kayo doon sa accredited nurseries, and the best organization na lalapitan po natin ay ang Department of Agriculture.
Okay. Thank you po, ma'am. So, siguro yun na po yung last. So, unfortunately, we are running out of time already. So, thank you for all your engaging questions po. And thank you po very much, Dr. Baltazar, for clarifying and patiently answering all of our questions. So, with that, we now move on to the next part of the program. Back to you, Miss Jessa.
Yeah, yan. So, thank you, Miss Shenny, and to our participants for their active participations, and of course to Dr. Baltasar for the additional insights during the Q&A portion. At this point, on behalf of the College of Agriculture and Food Science, we would like to present the Certificate of Appreciation to Dr. Miriam D. Baltazar for serving as the resource person on the webinar titled Identification and Genetic Characterization of the Philippine Coffee during the Coffee Webinar Series held on 21 November 2023 via Zoom. Given this 21st day of November 2023 at the College of Agriculture and Food Science, University of the Philippines, Los Baños, Laguna, Philippines. Signed by our Dean, Dr. Elpidio M. Agbisit Jr. Ma'am, baka meron ka pa pong message po sa amin, sa mga participants po natin. Walang message.
Ay, okay po. So thank you so much po for engaging into this webinar. Ano po? Yeah. My heart is full. Okay. Thank you so much, College of Agriculture, UPLB, my alma mater. Ano pa bang sabi ko? If you have questions po and kaya naman po naming sagutin, please send us an email. Pinapost ko po yung email address ko po dyan. But I can also be reached through FB. So pwede rin naman po kayo mag-send ng FB message. And you can send FB message directly to me or to the National Coffee Research Development Extension Center FB page. So marami pong salamat sa inyong lahat.
Yes, ma'am. Thank you din po for being with us today and sharing your expertise with us. So, and now, for the announcement, please stand by for our episode 4 Tuesday next week, November 28, 2023, with the topic R4D Initiative in Coffee and Future Prospects, to be presented by Dr. Calisco M. Protasio, adjunct professor from the Institute of Craft Science, CAFS UPLB. Also, let me take this opportunity to thank the people who made this event possible, to the CAFS Extension Committee and to the Ad Hoc Committee. Also, please give us your feedback for today's episode. You may access the evaluation form by scanning the QR code on the screen or through this link, which is also posted in the comments section. Also, please follow us on our official Facebook and Facebook page, UPLB College of Agriculture and Food Science, and our YouTube channel to keep you posted on the upcoming events. And now, for our last part of the program, to formally close this webinar, may I call the Station Manager of the La Granja Research and Training Station, Ma'am Eva C. Ginabo. Ma'am Eva, ikaw na po.
Okay. Good morning po sa inyong lahat. This is Eva Guiñabo from La Granja Research and Training Station in Negros Occidental. Thank you very much, Miss Sheni, Miss Jessa, Ma'am Doc Sherry, especially Dr. Baltazar, and the participants. Dr. Baltazar, thank you very much for your time and knowledge for today's episode. And for the participants, I really would like to thank you all for your very active participation and the very significant questions that you raised during the Q&A part of this webinar. Before I proceed, I would like to share with you the quick learnings that we've got siguro pareho din tayong lahat that Doc Miriam had shared to us. No, ang napulot ko doon ay mayroon yung 4 pa lang sinasabi nating varieties are not properly called varieties but types. So we have Robusta, we have Excelsa, we have Liberica, and Arabica. So now, safe pala to say 4 types. Thank you, Doc. And of course, as she also related to us, there are differences, there are similarities among these groups. But for those who are not really drinkers of coffee, coffee pa rin yun, di ba? Pero as Doc Baltazar said, she prefers Liberica, Excelsa, and Arabica. So for those who are coffee lovers, yun. Take it from the experts, from the expert, I mean. And of course, her topic also told us or also had shared to us that these 4 groups of coffee are adapted to, they have adaptability to different locations. So those are the 4 that I have picked from her sharing to us. But before that, let me also share with you, but before I really say the message that I have to share with you, I also would like to share with you some trivia on coffee that I've researched on, and I would like to thank Hills and Valley.ph Coffee Statistics Philippines. And share ko lang na I have learned from there that our country, Philippines, is the 14th largest coffee producer, and 95% of the producers are small farmers, meaning they only have less than 5 hectares of their land. And we are challenged because in the roadmap that they have identified, there will be a compound annual growth rate of 3.5 in 2021 to 2025. And we are the second largest coffee consuming country in Asia, because every single Filipino is consuming 2.4 cups of coffee a day, or 3.4 kilograms per person annually. What makes this webinar very important to this information that I am sharing with you? I would like to pick up yung importance ng araw na to, yung na-learn natin for us Filipinos. Wala na tayong way, but we need to really gene bank this 4 types of coffee, because yun nga, we are one among the largest producers, and even the consumer, nakita naman natin sa graph, mas marami tayong iniinom. We consume more than we produce. And small farmers, they are the most important stakeholders of this industry, of this commodity. And as educators, researchers, and extension professionals, and the issue of climatic change, we are challenged with this topic. One, I would like to share with you that this webinar opened opportunity for us to do more research on breeding and genetics, disease control and prevention, climatic adaptability. For those who are business enthusiasts in coffee, potential food enthusiasts, this is very important too. And for extension workers and educators, that this topic is very much timely. It's very much significant for our coffee growers to know all these things. Kasi nakikita natin na ang mga producers natin are really deprived of really proper information on production. Tayo naman hindi naman tayo, siguro kung hindi ka lang talaga coffee drinker, I mean, okay lang sa atin. But the productivity, the taste, and the quality of our product, of our coffee product are very important. So for the next episode, I wish you well. I know that there will be more enriching topics in the next episode, and I would like to give you good luck. And on behalf of CAFS, with the lead of our Dean, Dr. Elpidio M. Abis Jr., our Associate Dean, Dr. Lilia, or Doc Lilia, the CAFS Extension Committee, which organized this webinar series. Thank you very much. And we know, or every one of us really feels the breeze of the Christmas season. And I wish that every one of us ends our day sipping our very hot very strong, but very smooth and fine tasting coffee. Magandang umaga, maayong aga from Negros Occidental. Thank you.
Thank you po. Thank you po, Ma'am Eva, for that wonderful message and sharing the highlights for today's episode. So again po, scan the QR code flash on the screen or through the link posted, which is also posted in the comments sections. And once again, please stand by for our episode 4 of the Copy Webinar Series. Muli, maraming salamat and see you and kita-kits po. Kita-kits po. Thank you po.
Thank you.
Thank you.
Thank you po.
Thank you, Doc Miriam. Thank you, Ma'am Leah.
Thank you. Thank you very much. Okay po. Thank you.
Bye, Jess. Thank you.
Bye, Tishen. Thank you, thank you. Thank you,
Thank
everyone.
you, Jessa. Thank you, Jenny.
Thank you, CAFS.